Hi, I'm Jenny Kay, and I'm currently a postdoc fellow in the Engelward Lab at MIT. Right now, I conduct and communicate research as part of the MIT Superfund Research Program, but this year I'll become an independent investigator with the nonprofit Silent Spring Institute. My goal is to identify strategies for mitigating disease risk by researching environmental and physiological factors that impact the development of mutations and cancer. Mutations are key drivers of cancer development, and they are usually caused by damage to DNA. DNA can be damaged through a multitude of common sources, including external insults such as chemical exposures, as well as internal processes like inflammation. All organisms have evolved mechanisms to prevent mutations by repairing DNA damage, but they're not always perfect, so sometimes mutations happen anyway. One of the most toxic forms of DNA damage is double-strand breaks in the DNA backbone. Some DNA damaging agents directly cause double-strand breaks, but other types of DNA damage can indirectly lead to double-strand breaks as well. Homologous recombination, or HR, is a highly accurate pathway for repairing double-strand breaks, but on rare occasions, even HR can lead to mutations. Since many types of DNA damage can lead to double-strand breaks, the Engelward Lab developed the radar mouse, which detects mutations derived from HR. With these mice, we can identify conditions that cause mutations, which helps us to understand the potential for those conditions to cause cancer. For example, in this radar mouse pancreas, you can see fluorescent cells that have undergone the radar mutation. We can see not only how often mutations are happening, but also whether those cells go on to divide and produce clusters of mutant cells that may eventually become cancer. As part of the MIT Superfund Research Program, we've been studying mutations caused by the probable human carcinogen N-nitrosodimethylamine, or NDMA. NDMA contaminates the groundwater in the town of Wilmington, Massachusetts due to ineffective containment of millions of gallons of hazardous waste from the Olin Chemical Superfund site. In addition, NDMA has recently been found to contaminate common drugs in very high levels. As a result, many millions of people have been exposed to NDMA and may face serious health consequences. We have shown that NDMA-induced DNA damage causes an enormous increase in the number of mutations in the liver. This is especially interesting because NDMA causes base lesions, which are usually associated with point mutations and are repaired by pathways besides HR. Nevertheless, if those pathways can't efficiently fix the damage, then double-strand breaks occur and HR is activated. The ability to repair initial DNA lesions from NDMA has a massive impact on whether exposed animals will eventually develop mutations and cancer. In addition to my research within our Superfund program, I also lead our Research Translation Core, where I facilitate communication about our multidisciplinary program's diverse research projects to government agencies, other academics, affected communities, and the general public. As a new investigator, I'm excited to take more leadership in research and communication and help others strengthen their public health impacts as well.